Hi, I'm Bianca Sclavi. I'm a Director of Research at the CNRS. The main focus of our research group is to determine how bacteria adapt to different concentrations of antibiotics. And the idea is that when they encounter sublethal concentrations of antibiotics, they will change their growth. And they will continue growing, and they will, in addition, use this adaptation to be able to better survive when they encounter higher concentration antibiotics. So this is really important when you're trying to develop new therapies. They have an original way at looking at things, I guess, in that way. Usually people look at adaptation of bacteria by looking at genetic networks, for example, and focusing on a specific which genes changes when. We focus more on global, how different processes are connected to each other. So some people focus on DNA replication or cell growth and division or gene expression, and we try to, by this global approach, see how these are connected to each other. So this is quite unique. Not many people do this. It's quite a fundamental approach to science. So there's no direct application in the medical field, but by understanding this level of adaptation, we can, other people can use this information to develop new antibiotics, and especially antibiotic combination. I was always interested in quantitative biology, to have a quantitative measure of biological systems so that we can use this data then to make mathematical models of biological systems. And so in order to measure quantitatively the gene expression in live cells and in live bacterial cells, we needed very good control of the environment. That's how I started to look at in vivo cells in the microscope. And I first started with agar, but that has a lot of limitations and you cannot go for a very long time. So then we decided to switch to using microfluidic devices where we have a constant environment and control environment for a very long time. We can control it and we can change it, and things that we cannot do with agar. In the early 2000s, so almost 25 years ago now. Yeah. Here in this lab for about 8 years, but we started much earlier. In about 2004, we had started a collaboration with colleagues in the UK, Cambridge University. So we began the work in Cambridge. And so one of my PhD students went there to learn how to do it, and then she brought the knowledge here, and we started to do it here in about 2019. We started using agar. The limit there is that bacteria can only grow up to a certain point, and they form a colony, and then the colony will start growing in 2 dimensions, for example, and that we cannot longer do single cell measurements. And then once the colony fills the field of view, then the experiment is more or less finished. So we decided to switch to microfluidics to also control the environment and change the environment. And so we started with our colleagues in Cambridge University just using pumps, but we really liked the idea of being able to program the switch and the idea to switch between different concentrations, or the flexibility that a system like Elveflow could give us. And there was a colleague in Saclay, in the University d'Orsay, who is a chemist actually, and he's the one who showed us Elveflow for the first time. He really liked it, and he really liked the plug and play. We tested it with him first, and then so it's always a word of mouth. It's a lot of how decisions are made in our field, I think. I remember when we started using it. I was also with my PhD student at the time, And I think one person from Elveflow came here the first day and showed us how to do it. And mainly after that, it was just actually from our side to made adjustments and changes, and we had to be careful about filtering the medium and things like this. More or less, we were still using the same protocol that we used at the beginning. It's like 70% of our work is done with microfluidics. I mean, it's very time consuming. It's not easy experiments to do, but the amount of data that we get in the end is worth it. But yeah, it's a very powerful approach. It is difficult, but if you spend some time at the beginning to talk to people and to take advantage of the resources that are available now, it's actually not that hard. The nice thing is that now, since we've been using it for a long time, there is a lot of resources, a lot of experience on this field. So I think it's much easier for people who start now. And it's user-friendly. It's much more user-friendly now. Don't be afraid to start, but it takes time and you have to have a lot of patience. Okay, so like the experiment we are doing today, we set up the PDMS chip, then we inject bacteria in the chip, bacteria that are growing in exponential phase. The bacteria enter the channels, and to do this, we need to be able to control the flow rate, to make sure that there are no bubbles in the system, especially no bubbles in the small channels where the bacteria will enter. And then, once the bacteria go in, we let them adapt to their new environment, and we let them grow. And we need to acquire data before the injection of antibiotic to see the difference. So we let them grow there for about 6 hours, and then we switch the growth medium using the MUX device. And for now, we're just switching between no antibiotic and low concentration antibiotic. But in the future, with the MUX system, we can also switch between up to 8 different concentration of antibiotic. Today's experiments, we're just going to go from 0 to low concentration. Then the next step is to go from 0 to low concentration, and then add a high concentration, like lethal concentration, and see how this first adaptation step has helped bacteria survive. If we add the high concentration right away, there would be a very different response. So this we could not do if we didn't have the MUX system and the fact that we could program. Yeah, so we have collaborators who work on other bacterial species who come to us exactly for that, to do experiments on their species. Sometimes the chip needs to be modified a bit because of their different sizes, different shapes. But yeah, we're working on different species as well. Thanks to this system, we've been able to work on 2 main projects. I've talked about the project on adaptation to antibiotics, but another key project was to study the cell cycle of bacteria. And bacteria are not easy to synchronize. If you synchronize the cells, you get a lot of artifacts. So the only way to really study the cell cycle is to study the bacteria, the cells one by one. And the only way to do it is in a microfluidic device, a microscope. And so, thanks to this approach, the former PhD student was able to look at how gene expression changes as a function of the DNA replication status of the cells in the cell cycle. And this actually was just published recently, so it was a really nice work. For our application, the MUX is the biggest advantage compared to the syringe pump. And the fact that we could, in theory, switch between several different growth media. I have colleagues who try to mix different bacteria together and see how they work together. And so that is a new level of complexity. So the injection of bacteria in the device is something that is still not. We do it by hand now, but it should be automated, and something that would be the next step. Yeah. It's really easy to set it up, and it's really nice to be able to control and to have a visual of the system, and the fact that you can see the flow rate change in real time on the screen and the pressure. So it's easy to use and it gives you very good control. So we are very happy with it, of course, and also with the customer support. Whenever we had some problems, we wrote an email and we got an answer right away. If the person who was answering email didn't know, they sent us to an expert that came back for the technical side of things and also for the programming of the timing. So yeah, no, it's been really great. Yeah.